behalf of the Department of Bioengineering at the University of Pittsburgh, it is my pleasure to interview Professor Jim Collins of MIT, the Harvard Wyss Institute of Biologically Inspired Engineering, and the Broad Institute. Over the past 30 years, Jim has become one of the most accomplished biomedical engineers in the world and has made critical contributions in fields ranging from system biology to biomechanics. Above all, he's most likely recognized as the founder of synthetic biology, publishing one of the key seminal papers in the field more than 20 years ago. And in the last 2 decades, his research group has continued to drive the field, providing key discoveries and technologies ranging from synthetic gene networks that elucidate the fundamental principles of biophysics in the natural world to cutting-edge diagnostics for the detection of Ebola, Zika, and coronaviruses. Jim's been recognized with numerous awards, including the Rhodes Scholarship, MacArthur Genius Grant, and the NIH Pioneer Award. His accomplishments have led to him being part of the exclusive club of innovators that are members of all 3 U.S. national academies, those in engineering, science, and medicine. Personally, I was a postdoctoral researcher with Jim between 2010 and 2012, and I'm now a tenured associate professor here at the University of Pittsburgh. It's clear that Jim provided a springboard for my own professional success, and it is my pleasure to interview him today. With that, thank you, Jim, for your time today.
Well, Warren, thanks for the great intro, and it's always a pleasure to talk with you.
Well, thank you. Let's go right into it. So you're widely considered one of the founders of the field of synthetic biology, starting with your publication of the toggle switch, a bistable gene network in Nature in 2000. How did you and your co-authors get the idea to engineer a bistable network in E. coli? And why was that an important achievement?
We did not come from molecular biology. So in the 90s, my lab was primarily focused on applying nonlinear dynamics and control theory to whole body dynamics, studying how people walk, run, maintain balance, as well as organ dynamics and neuronal dynamics. I benefited at the time from the fact that the chair of the Department of Biomedical Engineering at Boston University, which is where I was a professor at the time, was Charles Cantor, who had been a PI of the Berkeley genome effort. And our dean of engineering was Charles DeLisi, who had actually conceived the genome project as a program officer of the Department of Energy. And in the mid to late 90s, the genome project was the project in the US life science community. And most bioengineers were outside of that whole space. We were like kids looking in on a candy store trying to figure out, could we play? Charles both thought that there was an opportunity starting around 1996 for somebody like me with a background in nonlinear dynamics and control theory to start contribute to the project. And they introduced me to folks like Eric Lander, Lee Hood, and others. And Eric and Lee, encouraged me to think about how we as bioengineers with various control techniques and approaches from nonlinear dynamics and statistical physics could begin to reverse engineer natural networks. So these were early challenges that helped launch what became systems biology. And we began thinking about how exciting that could be to use effectively machine learning to infer networks. And Eric and Lee and others clearly said, we have a challenge because we're getting these sequenced organisms with their parts lists expanding, and we now need to figure out how they work together in networks and pathways, and we need these approaches. We took on the challenge, began to dig in, and quickly realized the data sets were not yet available to reverse engineer. Microarrays had just appeared. There were only 7 publicly available data sets at the time. And so we ran quickly away from that problem, but had been sufficiently intrigued on what engineers could do to then say, okay, and instead of reverse engineering, could we forward engineer? So could we build networks from the bottom up? And it was the flip of what became systems biology, and it became then the dominant approach in synthetic biology. And we began to explore what could you do? What networks might make sense to build? And we had 2 criteria. One is the network had to be interesting enough in its features to be worthy of analyzing and building, but simple enough that we could analyze and build it. And our starting point was really going to be mathematical modeling, both analytics and computation. And part of the motivation there came from neuroscience. So I came up in part in the math bio community in the 80s as a grad student, and an absolutely fantastic, close-knit, very supportive community. But most of the efforts there were really focused on the model as the end goal and not of applying it to an experimental system to test its validity to gain insight. But in the early 90s, colleagues such as Nancy Capel, Eve Marder, Avis Cohen began to explore very small neuronal systems and test mathematical models against them. And in fact, a prime motivation for us to try to then build a small circuit, which became what is now the genetic toggle switch, was to see if we could have a platform to validate mathematical models of gene networks, which were in principle similar to mathematical models of neuronal networks. We, being largely a translational lab, also immediately recognized that these bistable switches, such as toggle switches, also had significant biotech potential in the context of industrial biotech, as well as gene therapy and cell therapy.
Well, so, I mean, I think I learned a few things in that response that I never knew. So I know that you outlined that you were in this field in math bio, doing human biomechanics and gait, and you had clearly cutting edge achievements there. To move in these new directions with Charles Kanner and the synthetic gene networks that you're describing, and these mathematical models of new systems and things like E. coli. I mean, wasn't that a significant risk to leave something that almost leave something where you were putting out cutting edge papers to kind of go into an area where you had never been before? I think many people are tempted to stay with what's working and kind of continue to build that first chunk of work.
You know, it's an interesting question. So to be clear, I didn't think of it as a risk at all, in as follows, in that with that initial effort to the genetic toggle switch, it's not that we were abandoning our earlier efforts. It wasn't that we were going to transform the entire lab. It was an exploratory project that was not unrelated to what we had done before, and that we had done a good number of ODE models of different coupled systems for representing neuronal nets. We were expert at applying stochastic processes to biological systems. So for me, I initially trained in physics, then moved into biomedical engineering. Never viewed myself as being limited to a particular system or space. And even in the context of nonlinear dynamics, we were fairly broad on ranging from neuronal control to more traditional biomechanics, animals to humans, gait, balance, jumping, weightlifting. So you had a broad scheme there, and nonlinear dynamics then, which became our prime focus in the 90s, gave us huge latitude to apply techniques to different systems. And it became what we were excited about. Now, a challenge is when you try to go into a new field in academia, you're an outsider. And our fields, our communities are tribal in many ways and can be very suspicious of outsiders. And we always tried to leverage our entry points from positions of strength and tried to make some inroads into the community, but still kind of coming in as motivated amateurs to bring new ideas. So it wasn't a huge risk, in that we probably at the time maybe had a 20-person lab. So to have a grad student and myself go after it, and it was an unfunded project, the genetic toggle switch, was an interesting, I wouldn't say it was a side project, it was an area that we thought had potential. And as the results came in, we saw, boy, this could be interesting. And I began recruiting additional graduate students. And over that next 2-3 years, probably brought on 3 or 4 of that first generation of grad students. And over the next 6 years or so that we moved the lab more and more toward molecular biology, synthetic biology, and actually then returning or going back to challenges in systems biology and beginning to abandon the neuronal network and biomechanics work of the 90s that we've done.
Well, this is really some great insight into how this field started and the process of expanding. And I'm sure there's a lot of lessons for our viewers there. Now that it's been a couple decades, I know you've mentioned in other lectures that the field has gone from an emerging discipline to more of an established discipline. And I know along the way, technologies like RNA interference and CRISPR genome editing have come out. They've won Nobel Prizes, and some of that technology has appeared in the synthetic biology that you've done in your lab. And I guess the question that comes to mind is, does this mean that synthetic biology is a field where it's technology agnostic? And in other words, is there a defining set of approaches and design principles and so forth that makes synthetic biology something that stands apart from a particular achievement, as breakthrough as the achievement might be?
Yeah, it's an interesting question. I think synthetic biology is more about approach, attitude, and philosophy than a specific technology or application. So I think RNAi and CRISPR were fantastic developments that really leveraged basic science insights toward the applied side and became a kind of a toolbox, a platform that transformed various fields. Induced pluripotent stem cell work by Shimanaka was a similar thing. In synthetic biology, it's really the idea that we can bring engineering principles into molecular biology, really enabling us to engineer circuits, engineer components, to create programmable elements that enable a living cell or a cell-free system to have expanded functionality, opening up applications. And it is a bit of a big tent, but allows many, many different technologies to come to bear. And so it's much more an approach than a specific substantiation of that approach that defines the field.
So that leads me to my next question as we're defining the field. As a junior contributor to the synthetic biology community, I go to conferences, I see pioneers like you speak, and it seems that some people are very driven to define what synthetic biology is, and as the field expands, to attempt to draw lines between what synthetic biology is and what it isn't. I'm wondering if you see value in that. Is there a clear distinction, or is there something more going on with synthetic biology becoming a field and an established field? Is it a natural process that more and more things will be part of the field?
Yeah, it's interesting. You know, I think academia and specifically academics in academia like to argue about definitions of terms and fields. And I remember when the Department of Systems Biology was established at Harvard Medical School, the chair of the department, in fact, said he didn't know how to define the field, which I found a fascinating comment. I like to think that synthetic biology is quite a broad field, and I like to think of it as a very big tent. Having said that, I do think it's important to draw distinctions as to what counts as an engineering principle. So in the 90s, before synthetic biology appeared, the term genetic engineering was used quite commonly to describe efforts in biotech. And there, the idea really was used to describe that we could take a gene from organism A and put it into organism B and to produce its associated protein at scale. That led to many debates within biomedical engineering departments around the country, which had not yet shifted into the molecular space, that the biologists in this case have co-opted the term engineering inappropriately. And I was one that agreed that I didn't think it referred to engineering, in that if I changed the color of this room, I didn't engineer. And the example I make is that genetic engineering would be as if I'm, for example, changing the color of a light bulb from red to green. Synthetic biology is focused on creating the circuitry to control the functionality of that light bulb, whether it be a toggle switch that could allow you to flip that light on and off with a mechanical stimulus or a temperature stimulus or chemical stimulus, that could enable it to come on and off periodically, that might enable it to respond to time or to temperature or movement in the room. And so synthetic biology gives you that element of control. So now as you go beyond, it becomes intriguing as lines get blurred on kind of older school and more biological manipulation versus true engineering. And I think it can lead to some interesting debates and discussions. As with many, the field will probably eventually become more and more absorbed into even more established fields, and or new things will pop up that try to delineate specific spaces and topics.
Well, that's great. That's definitely some insight for our viewers. I think I'd like to shift gears a little bit and maybe talk a little bit less about those of us who are in the field or other biomedical engineers and talk about the general public. And one thing that often happens when synthetic biology is mentioned, I mean, sometimes even with other engineers or scientists, is a concern about safety. Why do you think people are concerned?
I think people are concerned because the idea that we're going to manipulate a living cell or an organism or a biological component and introduce it into a patient's body or introduce it in the environment raises concerns of, will this lead to untoward consequences? Will it lead to health challenges? Will it lead to environmental challenges that were unforeseen? So, for example, in this vaccine rollout, I have a number of people who are very concerned about the synthetic mRNA vaccines, which I think are an example of engineered components that would fall under the banner of synthetic biology. And it's a new technology, so people get concerned, is this technology going to be safe? And I think these concerns are important to air and voice, and we as a community need to do as good a job as possible to firstly address the safety issues in our labs and associated schemes and share what we know and put in appropriate safeguards, be it biocontainment and or safety switches that can eliminate what we're introducing as needed.
So I know that your lab has had several projects focus on putting in safety switches and into your synthetic biology outputs. So clearly it's something that's immediately important to you. I guess the question I have is, what degree should we involve non-engineers, non-scientists, such as maybe government officials or academic or think tank bioethicists or people like this in monitoring synthetic biology and to ensure that nothing unsafe happens?
So I think it's important to have discussions with folks, like one could say, outside the field. So involved with ethics, legal concerns, philosophy. I think it's a very good idea, and I think the field has engaged those communities from the very beginning. I critique the field a bit in that I think many of our earlier research meetings got too involved with such efforts, and I think it's important to have the research as a prime focus. It's the research conference, and then on the ethics, and to continually kind of blur the line between the two. So I've had ethics talks on either side of talks that I give at big synthetic biology meetings. I think becomes problematic in that they really are addressing different skill sets, different expertises. But having said that, I think particularly as we move, so to go to an earlier point you raised, where I speak about the field moving from an emerging field to a maturing field. Part of the maturing is that we're increasingly seeing synthetic biology technologies, platforms, products being introduced into the market and are tested for clinical application. And there we have in place, in many cases, good safety schemes, whether it's the FDA or the USDA or the EPA. And I think we need to engage them in all processes and serve to educate officials along with our young folks of what are the potentials and what are the potential benefits and the potential hazards.
That's a great answer and actually opened my thinking up quite a bit. I'd like to shift gears again from safety and actually ask about a new direction, somewhat new in the field of synthetic biology. I know over the last decade, your group has made significant strides in many areas of synthetic biology. And one of these is an area known as cell-free synthetic biology. Could you tell us a little bit about how that differs from maybe synthetic biology done in other ways with cells, and what advantages there might be for using this approach?
Yeah, so cell-free synthetic biology has emerged as a very active subfield of synthetic biology, and we are participating in that subfield. The general notion is that you can open up a living cell and remove its machinery and play with that machinery in a petri dish or a test tube or on a different substrate. That machinery could be DNA, RNA, ribosomes, other molecular machines, molecular components, molecules such as ATP, nucleotides. Cell-free systems have been utilized in molecular biology for over 6 decades. So Marshall Nierenberg and his colleagues actually worked out the RNA code using cell-free systems. The difference, obviously, with a cell-free system versus a cell-based approach to synthetic biology is the lack of a cell. So you're not working with a replicating or metabolizing cell with its genome and many other processes underway, which can lead to simplification of interactions or reduction of interactions that could lead to unexpected results or play with the output of your system. That opens up the possibility that you can better study your system in isolation and characterize its dynamics. And we said that cell-free systems, beyond providing a platform that gives you greater control over your engineered elements, it also provides means that allow you to take synthetic biology out of the lab. So with Keith Pardee and others in my lab, we pioneered the idea that you could freeze dry cell-free systems onto paper, onto other porous substrates like clothing, or as pellets that then open up possibilities for paper-based diagnostics, wearable diagnostics, portable on-demand biomolecular manufacturing, as well as educational kits. So this is enabling now the power and diversity of synthetic biology to have greater reach, where, for example, with the freeze-dried elements, you don't need to have refrigeration. So you can freeze-dry these schemes, keep them at room temperature. We've shown out to about at least 2 years without significant loss of activity, opening up field-based uses as well as utility in low resource settings where you might not have a high-end refrigerator. And again, this is serving, I think, to extend the reach of synthetic biology to different parts of the world for different applications.
And to follow up on that, because there's no cell, I presume that this also means that there's no chance that anything that's, back to the safety question, that nothing can replicate, nothing can infect somebody, that these cell-free technologies would then be incredibly safe. That's accurate?
They would be, in general, safer than your other schemes. You still have biological material. You might have transposon elements or plasmids played in these cell-free systems that still could represent some schemes, but they are also safer. You can also envision you could play with viruses in a cell-free setting, and viruses themselves are operating and can function outside a cell. They need the cell to replicate, but by eliminating the replication machine, or the machine that can continue to replicate, it significantly reduces risk of untoward escape that would be problematic.
Great, thank you for the clarification. I want to ask about another part of your cell-free work that actually ties into something bigger. So for our viewers, I didn't mention this in the introduction, but you have also won significant recognition for your teaching efforts and commitment to student education with departmental and even the university-wide teaching award at Boston University when you were previously a faculty member there. Synthetic biology has been recognized as a technology not just for advancing medicine and other bio-dependent industries, but also as a platform technology for outreach. And your institution hosts a worldwide SynBio competition. And I've noticed that you have developed, and you've been part of a group that has developed the BioBits platform based on cell-free. I'm wondering if you could tell us a little bit about that and how that might make an impact in educating the public about both synthetic biology and engineering principles in general.
Yeah, so this was a collaborative effort with Mike Jewett's lab in Northwestern. And in my lab, Ali Wong, who was then a PhD student, led our team. And in Mike's effort, Jess Stark, then a PhD student, led his team. And we recognized that these cell-free, freeze-dried systems could open up the possibility of creating educational kits for young folks to learn about synthetic biology and biotech. And the motivation was recognizing that hands-on experiences, particularly amongst the young, are great ways to excite them about a field and to introduce them to a field. And I think of chemistry kits, for example, from my youth. It remains the best gift I ever got when 7 years old from my dad's mom. One of these old chemistry kits that had, fantastic elements of combustibles, explosives, poisons. And I did everything in the book and everything off the book that I could think of. And it gave me hours and hours of fun and exploration. Nothing like that exists in the biological space. And you can envision parents really being concerned if their daughter's trying to grow her brother's stem cells in the kitchen. These cell-free freeze-dried systems can be operated in, frankly, safely at a kitchen table or in the basement or in the garage, enabling young folks to explore properties of transcription and translation, enabling them to play around with enzymatic reactions to build synthetic gene circuits, to build sensors that can enable them to test DNA from fruits or from animals. And it opens up this possibility in an inexpensive manner to allow kids both to explore at home, but also in their classrooms, where most middle schools, most high schools don't have high-end biology labs. And this would really help train and excite this next generation. And Ali Wong joined Mini PCR Bio that's looking to commercialize these kits, or is commercializing these kits. And what Mini PCR Bio did in the midst of the pandemic was fantastic. They actually donated 250 of these kits to the Boston school system to enable young kids to participate in remote learning from a hands-on standpoint while school was not actually in person.
Wow, that's fantastic. You brought up the pandemic, and I think I've got maybe one question to close on that ties in a little bit of your other work. So you've also been a pioneer in understanding the emergence of antibiotic resistance. And just as people like you and others predicted that worldwide pandemic was a possibility, a virus-based pandemic, the coming potential of running out of antibiotics is something that we often hear about in society. It seems that we're constantly told, stop over-prescribing antibiotics to patients, stop feeding factory farmed animals antibiotics so we can have them live closer, and things along these lines. That doesn't seem to be working. It seems like antibiotic resistance emergence, I should say, is a continuing problem. How likely is that a problem to come? Is that going to be the next global health crisis after we get out of this pandemic? And is there a role that maybe synthetic biology can play in fixing that? That's a big question. If you could answer some of that, that would be
happy to. I do think it's a crisis. It's a global crisis that is here and is going on even in the midst of the pandemic. And just to share, I think the challenges of bacterial infections are not independent of the current crisis. So 1 out of 7 patients who are hospitalized with COVID have a bacterial co-infection. 50% of the patients who die from COVID have a bacterial co-infection. And you look back to the Spanish flu of a century ago, which preceded the introduction of antibiotics, it was as deadly as it was primarily because of bacterial co-infections. We are in a crisis around antibiotic resistance, and a number of groups are raising attention to it, but the public hasn't responded as well. I wouldn't say that efforts in antibiotic stewardship aren't working, it's just they aren't working as well as we'd like. So I expect without them, we'd see even further increased rates of resistance. Unfortunately, those stewardship programs are not yet leading to major development and discovery efforts, which we need. I think synthetic biology and broadly bioengineering can play a very big role to advance this. Synthetic biology, I think, offers new means to introduce antibacterials, be it in the development of engineered bacteriophages, the viruses that go after bacteria, the better engineering of antimicrobial peptides, the engineering of living diagnostics and therapeutics, so engineering other bacteria, for example, to detect and treat bacterial infections. And we did so for treating cholera, a nasty bacterial infection that's been dealt with humans for centuries. I think synthetic biology offers also tools that enable one to gain insight into bacterial infections. So we use the toggle switch in engineered bacteria to report out on what was happening at infection sites and actually challenge general thinking about what underlies a tolerant infection. Broadly, I think bioengineering efforts utilizing AI and deep learning, potentially in conjunction with synthetic biology, will be a way to help get us out of this challenge. And the numbers are stunning. I think it's about 1.5 million people die each year from resistant infections. That number is projected to grow to 10 million per year by 2050 if we don't address this crisis. And part of the challenge is that it's just kind of simmering in the background, that I think all of us, anybody listening to this interview, have a loved one or a friend who died from a bacterial infection, typically probably one that they caught in the hospital. But it happens relatively quickly. Rarely is it a long-term issue like cancer or neurodegeneration. And so it doesn't rally that strong family response. So of note, there is no major foundation really focused on antibiotic discovery and development. There is no month dedicated, there is no day, there is no march, there is no road race, there is no color, there is no ribbon, there is no celebrity endorser behind it. And as a result, the public just isn't as engaged on this. Now, I don't know if it's the next pandemic or the next crisis, but I think it's a crisis that's occurring on a global scale that needs increased attention and needs new and more resources.
Well, thank you for that answer and that insights into this problem. And I just want to thank you for your time today, Jim, on all of these topics. It's really been fantastic to hear your insights from the basic science all the way to some major societal problems. And it's clear you're thinking about all of them. And just thank you for your time today.
Well, Warren, as I said, I always enjoy talking with you, and this was no different. So that was a great interview. Thanks so much.
All right, thank you. Bye-bye.